Good evening and welcome to Kopernik Observatory. My name is Drew Desker. I'm the director here at Kopernik, and it's great to have a nice crowd here this evening, especially considering the weather. But there's always a lot of good stuff going on inside here, and you're here to enjoy it. Let's see, by show of hands, who is here for the first time? Okay, good portion of you. Okay, or who hasn't been here since your 4th grade field trip? Okay, a couple of you, all right. The other question is, who here are members? Okay, almost the other half there. So what I'd like to point out is that Kopernik, we are a nonprofit, but we belong to what's called the Association of Science and Technology Centers. It's a consortium of over 380 other science centers that all share each other's memberships. So if you like this kind of a place, a Kopernik membership is actually a good value because you could go to Roberson for free, you could go to the Museum of Science and Technology up in Syracuse, you could go to the Intrepid Museum that has the Enterprise, the space, that sort of test space shuttle down there, the Franklin Institute down in Philly. And actually, a number of years ago, I took my daughter down to the Franklin Institute, and we paid just the regular admission fee for myself and just my daughter, it would have been more than the cost of a year membership. So if you like this kind of place, a Kopernik membership is a great thing to have. So we are actually celebrating our, just sort of closing in on our 50th anniversary. The construction started in 1973, but we opened in 1974. And earlier this year, we had a big gala down at the Doubletree Hotel. We actually had a Nobel Prize-winning chemist, Dr. Stanley Whittingham from Binghamton University, was our keynote speaker. And we are now about to announce embarking on a major campaign to expand this facility and expand our outreach. And so you'll be, hopefully over the next weeks and months ahead, learning more about it, and you have an opportunity to plug into that as you wish. On our website, we always keep it up to date with what's going on. We just released our first newsletter. We call it the Kopernik Constellation. It'll be sort of a quarterly update on what's going on here. But also, we tell you what's up next here. So tonight's program on bacterial biofilms is tonight's program. Vigilia 2024. So not everybody knows this, but again, Kopernik was a Polish astronomer. And the Kopernik Observatory and Science Center was actually started by a group of Polish immigrants and people of Polish heritage here in the area. And part of our bylaws is actually to promote Polish heritage. And Vigilia is actually the Christmas Eve dinner that is celebrated in Poland. And so actually on Sunday evening, we are having our Vigilia down at the Doubletree Hotel. You had to have reserved your ticket already, so maybe next year you can plug into it, all right? But then if you go up to our website here, you can go to, that's not really where I wanted to go, so let's try that again. Okay, here we go. Classes and programs, public programs. And so you can come over here to upcoming Friday nights and learn about what we do. So from March to mid-December, we do a public program on a wide range of topics, not just astronomy. And obviously tonight's program is evidence of that. Next Friday is actually a special thing. It's a special screening of a movie called The Star of Bethlehem. And it talks about there was actually a gentleman who was interested in what did the actual wise men see, around the birth of Christ. And so there's some software that emulates what we can see in the sky. And basically he took passages from the Bible and tried to map them into what was actually in the sky, over 2,000 years ago. And so you can investigate that. And then finally, you keep hitting the other button here. And then finally, we're going to close out this year with actually another Binghamton University professor, Tom O'Brien, out of the education department. It's going to talk about science and religion. a tale of two theories or evolutionary epiphany. And that will close out this year. In January, we'll have a single event, and in February we will have our Winter Star Party, where we celebrate Kopernik's birthday. Also, behind these chairs over here is our cornerstone that was thank you. It was part of the original building. And actually behind that cornerstone was a time capsule, which for those of you who are here, we actually have that time capsule in the back. We are going to re-intour the time capsule, including putting new material into that, so that in 2074, somebody else gets to open it and see what it is. So, and then our Friday night programs will start up again in March. So these Friday night programs I love doing because it's an opportunity for us to really continue our education. So especially all you younger people here in the building, your education doesn't stop at 12th grade or even at college. It really meant to be a lifelong effort. And we're not going to turn everybody into microbiologists or astronauts or engineers, but being an informed citizen is not a bad thing to do either. And so one last thing I'll sort of talk about is so we've been doing these Friday night programs for decades. And so back in 2020, we had to stop because of COVID. We couldn't put people in a large group like we're doing tonight. And so we said, okay, well, we'll just hold off for a couple of weeks until this COVID thing blows over, and didn't exactly play out that way for us. So after about a month or so, we said, look, we've got all these people lined up to talk. Why don't we do something about it? So we actually had them Zoom into our facility here. We then turned it back around on our YouTube channel. We now have over 13,000 subscribers. Now, I don't think we'll get 13,000 people tonight on our live stream, but what's great about it is that if you go to our YouTube channel, just go to Kopernik Observatory, and then so you'll see not only what we've got going on tonight, but you can see like last week we had black holes on Black Friday. The week before that we had a talk on CRISPR, the gene editing process. Then there was an astronomy, mythology, and the keeping of time. So if you missed a program, or if you can't get up here on a Friday night, you can just plug into our YouTube channel. So anyway, we are here tonight to learn about biofilms, and I have to say that I don't know very much about it, and I'm actually very much looking forward to this talk. And so we're very fortunate to have a world-class university and world-class educators here. And tonight we have three of them to present. So I'm going to introduce Dr. David Davies, Dr. Karin Sauer, and Dr. Claudia Marques to talk about biofilms. So, so let me switch you over here.
Okay, thank you, Drew. Is my mic working? Yeah? Okay, terrific. So today we're going to talk about, and I'm David Davies. I guess I'm the guy of the trio, so you can figure that out. So we're going to talk about bacterial biofilms, microbial communities that behave like multicellular organisms and impact every aspect of life on our planet. So before I even jump in, let me put a little bit of perspective on this. So the idea here is that, you're people, you've evolved, you're the result of 4.5 billion years of evolution. You think of yourselves as these higher organisms, but how did all of your tissues start? How did all of these components that go to making you up get to where they are now, right? And also, what are those original kinds of assemblages of cells doing today? So what we're going to do is actually talk about this. We're going to have a few examples of why this is important and look at something that's a little bit different than just talking about microbiology. All right, so without further ado, first I want to say that most people, and even other biology professors, sadly, consider that bacteria, and this is a picture of some bacteria, that they're nothing more than just a bag of enzymes. So there's not a lot of structure inside a bacterial cell. And these things are so small you can't see them anyway. So this is greatly magnified, of course, right? So, but there's much more to bacteria than just being a bag of enzymes and carrying out enzymatic activities. So bacteria can be found in a number of places, and I'll talk about basically across the biosphere where we run into biofilms in bacteria, but they have a number of properties that are very important. So they can communicate with one another. Bacteria can actually talk to each other by chemical communication. So one bacterium can say something, the other bacterium can pick that up, and then they change their behavior as a result of that. Groups of bacteria can act together in concert to do some kind of activity, and I'll talk a little bit about this later. They can make products. So, you know, we're running out of oil, right? So maybe not in our lifetime, but, or at least my lifetime, but maybe in your lifetimes. And we need to come up with solutions to create things like plastics other than relying on oil to do that. Well, bacteria can actually make plastics, right? And they do it without using oil. So we also have organic solvents like alcohols and butane and or butanol and other kinds of organic solvents that we use. Vitamins are produced by bacteria, antibiotics. Many of them are produced by bacteria. All are produced by microorganisms. In addition, bacteria can help higher organisms to digest our food. So you may not know this, but you don't digest your food. It's the bacteria inside your gut that are actually producing enzymes that they release and digest your food. And then you absorb the nutrients through your intestinal wall. They can coordinate their behavior, and they live in a universe we rarely glimpse, but one that impacts us every day. And we'll give you evidence of that in this talk. This is one of my favorite diagrams, and this puts my life and the situation with our understanding of biology into context. So here we have the tree of life. Most people are familiar with there being a tree of life with the different kinds of groups of animals and plants and microorganisms. And each one of these branches represents a different type of organism. And here on this anemic branch at the very bottom, this twig that looks like it's about to break off, are the bacteria. And then all of the rest of this tree is all of the rest of the organisms living on this planet. Trees, whales, insects, worms, right, are all what's on the rest of this tree. The problem with this tree is it's completely wrong. This anemic twig down here is not an anemic twig at all. This is the real biodiversity that exists on this planet. This is based on genetic analysis, where the genomes, that is the combined DNA from an organism, from all of the different kinds of organisms that are out there, has been sequenced and compared. And so all of the organisms on this tree, except for this twig, all of those organisms are actually just here. Just this part. This is you, this is an oak tree, this is a blue whale, this is a spider, this is a blade of grass. Everything that you can think of, it's your pet, right? It's the birds that are flying through the air. It's everything that you encounter that you know about is this small little bit of biodiversity. All of the rest of this are bacteria and organisms that are microorganisms that are related to bacteria that are called archaea. So you have bacteria and archaea, and organisms such as ourselves, we call ourselves eukaryotes. So that's where the real biodiversity is. And if you look at what's going on in the biosphere, most of what's happening is caused by microorganisms, caused by that biodiversity. Okay, so let's get into the topic area of this talk. So that is biofilms. And the question is, what is a biofilm? So the answer, this is the academic answer, is a biofilm is a population or community of microorganisms living at an interface and enmeshed within an extracellular polymeric matrix. Okay, so what does that mean? Let's break that down. All right, so population, what is a population? A population is a group of individuals arising from a single progenitor cell. In other words, all members of the same species, right? So human beings would be considered a population. Community is two or more populations. That means two or more species, right, living together. And a biofilm is a population or a community. So they could be a single species of bacteria that make up a biofilm, or they could be a series of more than two or more species living together. Microorganism, this is an organism simply defined as anything that's alive but too small to see without a microscope. So there's no genetic relatedness with respect to microorganisms. It's anything that's too small to be seen with the unaided eye. An interface, this is a location where two phases meet, so air and water, or water and solid surface. And then all of this is enmeshed in this thing that we call an extracellular polymeric matrix, and there's a real easy word for that you all know, and it's slime. So these things produce slime, and they live in slime, and the slime holds them together. So we have single species or multiple species living in slime on a surface. That's a biofilm. And these are just some pictures of some biofilms. Okay, so it's not always evident what bacteria actually look like. And so I show this image to show you a pin. So there's a pin. And we think of bacteria as being on the tip of the pin, the head of the pin. And so this is the head of the pin. This is an electron microscope image. And you can see the bacteria that are on here. And we magnify this again and again. So this is actually E. coli bacteria. And so this would be an individual organism. And all of these individual organisms, you could see this very sharp tip of the pin is actually flat on top, and it gives you an idea of just how big bacteria are, right? All right, so some basic facts about biofilms. So first of all, essentially all microorganisms are found within biofilms for the majority of their life cycle. So when I'm talking again about microorganisms, I'm mostly referring to these bacteria. So bacteria are usually found in a biofilm. And I tell people, generally speaking, if a bacterium is not in a biofilm, it's because it just left a biofilm and is about to enter another biofilm. It's in transition. And microbial biofilms occupy all niches where plants and animals are found. And they occupy environments that are also inhospitable to higher organisms like us. So waters that are too hot to live in, bacteria can live in and in biofilms, or too acidic, or too much salinity. So different kinds of environments, too dry even. Heat, cold, acid, salt, pressure, deep in the ocean, etc. So basically any environment where water exists, as opposed to being, and we'll see later that they actually do live in ice too, but where water exists, there are always microorganisms. And what are the benefits? Why do these organisms want to live in a biofilm? What do they get out of it? Why not just live on their own, floating around in some kind of nutrient? Well, if they're on a surface, living in a community or a population, that population or community can trap nutrients and help prevent detachment of organisms so they get sort of wafted into the air or into the water. So they get stuck onto the surface and can grab food as water passes by. The cells are protected from attack by reactive chemicals, so things like chlorine, which can easily digest or oxidize an organism. That slime that they're in is like a sacrificial layer, and it protects the organisms that are in the biofilm. Also, these cells are protected from grazing protists. This is things like amoebas and paramecia that are trying to eat the bacteria, but they can't get at them because they're in this biofilm, covered with this slime. They're also protected from attack by antibiotics, and they're buffered from changes in environmental conditions like pH and temperature. And maybe this is the most important to this audience. Biofilms are responsible for over 80% of bacterial infections in humans. So if you get a bacterial infection, it's almost certainly a biofilm. And you're going to learn why this is problematic. Okay, so just some pictures to give you an idea of what these biofilms look like. So sometimes it could be a single species, a population of bacteria at a surface that is acting as a biofilm. Here you see all of these fibers. That's actually dehydrated slime. So when you take the water out of the slime, it forms these little fibers. But once this is rehydrated, this would just be one big wet mass, and the bacteria are enmeshed within that. And then here is another image that shows multi-species. So oftentimes there are more than one species of bacteria that are resident together in a biofilm. All right, so I have a question for you. Here's a picture. This is an actual photograph of an object, and I'm wondering if you can tell me what this is a picture of. So the colored objects are different species of bacteria. Does anybody have any idea what this is? Like to venture a guess? The object. What is this a picture of? Yes? Digestive tract. No, that's a good guess, but that's not a digestive tract. Yes? A coin. Another good guess, but no, it's not a coin. Anybody? Yeah? A doorknob. These are all places where bacteria might be. One thing I want to point out is that typically a biofilm won't grow on something that's dry, like a coin, which also is antimicrobial. I don't know if you're aware that heavy metals in coins are actually there on purpose to suppress bacterial growth. And doorknobs, yeah, the bacteria can be there, but they're not really growing on that because there's not much food there. Any other? Yeah?
Inside of us?
No, it's not inside you. That's a good guess, though. Skin? That would be outside us, but no, it's not that either. All right, you give up? All right, you might, your mouth? It could wind up there. Are you ready? So you may be wondering, when you clean your plates after you eat dinner, are you removing the bacteria from your plate or adding more bacteria to your plate? So yeah, that's a kitchen sponge. And so if you learn nothing else, let me give you at least a little bit of sort of home ec wisdom. And that is, first of all, you're not supposed to use a sponge for more than about 2 weeks. Secondly, once you have a sponge that has gone 2 weeks, if you don't choose to throw it out, you can disinfect your sponge. The way to do that is to put it in a 2% chlorine solution and let it sit there for about 10 minutes, and then you stick it in the microwave. Microwave it, and then you can rinse it out with water and you can use it again. Don't say you didn't learn anything tonight. That's a microbiologist talking. Okay. All right, next. So one of the things that's interesting about these biofilms is that they actually are much more complicated than just these. Again, they're not a bag of enzymes. So these are individual organisms that are interacting with one another.
Pardon me, Professor?
Yes.
I just have to say, one of the people on the chat says, "Oh my God, I'm getting a new spreadsheet tonight."
There you go. Not a bad idea.
There you go.
Get a multi-pack. So one of the things that I mentioned to start out with was that biofilms actually look a little bit like a tissue, right? And our bodies are made up of tissues. So we have skin, and there are several layers of skin, and then you've got your intestinal lining, and you've got, you know, the different tissues that are associated with your organs, like your heart tissue and your lung tissue. All of these things are really not a lot different from different biofilms together. And I know it might sound crazy, and you've probably never thought about it this way, but all of the cells in your body that are able to divide actually have bacteria in them. And bacteria are responsible for all of the energy generation, or the majority of energy generation, that we have that keeps us alive. So you may not know this. You probably all heard of mitochondria, but a mitochondrion is actually a bacterium that's been captured by your cells. And in plants, chloroplasts are the same kind of thing. They're a captured bacterium. Actually, it's a cyanobacterium. It's photosynthetic. So plants get their photosynthesis from bacteria. You get your energy from bacteria. They evolved to do that in biofilms. And when these organisms are growing in a biofilm, they go through a life cycle. So they come to a surface, they start to build up biomass, and then eventually, after they become overcrowded, the bacteria will disperse away from the biofilm. They will leave and go form a new biofilm or join an existing biofilm elsewhere. The reason we care about biofilms particularly, and this is something relating to the work that we're doing at Binghamton University, all of our labs that are involved in biofilm research are actually looking at biofilms from a medical context. And the problem with biofilms is, and we call this the biofilm problem, that they're almost impossible to eradicate once they establish themselves in an infection. And there are 3 different mechanisms that biofilm bacteria use to protect themselves from being eradicated or killed. So the first is, and this is representing a biofilm colony, right? And there are many colonies that make up an overall biofilm. So penetration of chemicals that will kill bacteria, like antibiotics, is inhibited as the chemical tries to get into the depths of the biofilm. So the organisms on the outside might be easily killed, but the ones that are in the interior are often protected. The second thing is that bacteria know that they're in a biofilm. And when they're in a biofilm, they actually tune down metabolically. They grow more slowly, and they take up nutrients less quickly. So it's much more difficult to kill them, right? So let me equate that to something you might sort of be able to relate to. So if you're in an environment with poisonous gas in the air, if you hold your breath, it's not going to kill you. If you breathe normally, it will kill you slowly. If you exercise and breathe deeply, it will kill you fast, right? The same thing is going on with biofilm bacteria. What they're doing is they're breathing slowly. And so it's very difficult to eradicate them because of that. And then the third tier of protection is that biofilm bacteria actually turn on special genes that are associated with degrading antibiotics, that are associated with preventing uptake of specific antimicrobial agents. So they're protected from all 3 of these at the same time. it's one problem to solve one of these, but it's almost impossible to solve all of them at the same time. And this is the biofilm problem. When you have a biofilm infection, biofilm bacteria are protected by all three mechanisms simultaneously. And it makes it very, very difficult to eradicate them. So protection, reduced metabolism, and resistance. Okay, so biofilms generally include a variety of species that make up a community, and you can see here these are stained bacteria that are in a biofilm on the lower left, where each color represents a different bacterium. And then on the lower right here, the same kind of thing. And what we have going on here is one organism is actually cross-feeding with another organism. So these interact with one another. They communicate with one another, they cross-feed. The waste products of one are the nutrients of another. All right, so what I'd like to do to finish up my part of the talk is to go through fairly quickly a number of what we would refer to in our group as pretty pictures. So giving some idea of what biofilms look like, what kinds of environments they're found in, and kind of what they're doing. And then when I'm done with doing that, then Dr. Sauer and Dr. Marques will get up and give more detailed description of particular types of biofilms and why they're important. All right, so here's, and by the way, if you have any questions, just stop me. I'm more than happy to be interrupted. So this is a biofilm that's grown in a laboratory setting, and this is looking at the biofilm from a microscope perspective. And there's a substratum at the top, and the bacteria are attached to that, and they subtend or are dropping down from their attachment to the slide into, there's medium here that is flowing. So, and this is an image taken with this kind of a microscope. So that's kind of what a biofilm looks like under magnification. And you've got these different cell clusters like here, and that's typical of biofilms. So you see lots of different cell clusters like there were in the diagram I showed you previously. Biofilms are also found, here's another one of those, oh my goodness, slides. So this is, how many people here have contact lenses? Okay, so when you take your contact lens out of your contact case and put it in your lens cleaning solution in your case, this is a biofilm growing in the contact lens case, right? So you're taking them from your eye and you're putting them in there, and then the bacteria attach to your contact lens, and then you put it back on your eye. So you might want to think about making sure your contact lens case is clean. Okay, here's another picture of, these are bacteria on different kinds of microorganisms on a rock in an alpine stream. This is a Pseudomonas aeruginosa infection. That's a type of bacterium that you don't want to have infect you, that is on a voice prosthesis. So somebody had to have their voice box removed, and they have an artificial voice box we call a prosthesis. And bacteria have started to grow on this because there's no immune function from this piece of plastic. And so they can then colonize other parts of the body, and that could be very problematic. And then finally, I love this slide because this is really cool. These are root hairs from a plant. And this is a biofilm that is growing on the root hairs, and those bacteria in the biofilm protect the plant from pathogens that, or infecting bacteria that can get and kill the organisms. Did I just steal something you were going to talk about? Okay. Wastewater treatment. This is actually an area where we want biofilms. So you think about the sewage treatment plant, you're pumping wastewater into a facility that is trying to purify that water. That's done by using bacteria as biofilms to digest all of the nutrient that you think is disgusting, right? They look at as food. And when they're done, you've got clean water coming out. So these are the guys that mostly make our water safe. So they're not all bad. Then you've got some crazy environments. This one is really cool. These are tube worms. This is in a hydrothermal vent community, way, way, way down at the bottom of the ocean. And you've got these black smokers that are taking water that is deep subterranean water that is around 300 degrees centigrade. That's way, way higher than boiling at this temperature, right? So boil at this pressure. So this is under extreme pressure, so this water is not boiling, but it's very, very hot. And it has lots of nutrients. And it turns out that those nutrients can be used as an energy source for bacteria that are growing in this environment. But not just anywhere in the environment. They're growing inside these worms. These are tube worms. They're in the gut, which we call a trophosome, of the tube worm. And this is really cool. In order to bring, so they're actually growing on hydrogen sulfide, which would kill you if you breathed it. But that hydrogen sulfide is actually the energy source for these bacteria. And in order to bring the hydrogen sulfide from the overlying water to the bacteria in the tube worm produces hemoglobin. That's why they're red. So the hemoglobin binds the hydrogen sulfide, but this is the really cool part. Hemoglobin is made up of a protein, that's the globin part, and heme. The heme from hemoglobin, from this hemoglobin, is made by the bacteria. The globin part is made by the worm. So you have one molecule that is actually made by two individuals living together. And so those bacteria are actually feeding these tube worms on something that if you breathed in, it would kill you. And that's the basis of the food chain in this environment. Pretty cool. So hot, Hades, coming from the bottom of the earth. What's the opposite of that? Cold, right? So how cold do you think bacteria can tolerate?
As cold as it can get.
As cold as it can get. All right, so let's look at as cold as it can get. Antarctica, ice in Antarctica. And here we have biofilm bacteria growing inside ice in Antarctica. And what's going on here is they are photosynthesizing. There's some photosynthetic bacteria. They're getting light from the sun, and they're producing sugars, and the other organisms that they're growing with are feeding off of those sugars.
Are they producing heat as they're growing?
So there needs to be water. That's a good question. So there needs to be water in these fissures in the, I'll stop soon. She's looking at her watch going, oh my God, he's going forever. So yeah, they need to be producing enough either heat or antifreeze. So they actually produce chemicals that can lower the freezing point of water so that it is liquid where they're growing.
Would that antifreeze be glycol?
It's related. No, it's a protein. Usually they're proteins. So, but it has the same sort of impact as glycol, right? Another thing is like wood, right? So you know that termites eat wood, right? Did you know that termites can't digest wood? Do you know that? So there's a termite, and here's the gut from a termite, and it's in the gut that the wood is digested. But it's inside the gut that you have these ciliates. This is crazy, right? The ciliates are growing and crawling around in the gut, and then inside the ciliates are bacteria. And it's not the ciliates, it's not the termite, it's the bacteria in the ciliates in the termite that digest the wood. So the termite can chew it up, it can masticate the wood to make small pieces that the other protists, which are eukaryotes, can make even smaller, and then the bacteria take them and they digest them with enzymes. So wood, if we didn't have bacteria digesting wood, think about it. Every tree that has ever fallen would still be sitting on the ground. And that's a lot of trees. By the way, just so you know, the oldest forest on Earth, you're in the middle of it right here. So the first trees that ever evolved on this planet were in this area. One of my favorites, this is in the desert. So this is Dr. Sauer. By the way, we vacation together because we're married. So it's not entirely accidental. But what's really cool here, besides Karin, is these black and red stripes that you see kind of flowing down the cliff. That's something we call desert varnish. And desert varnish is not just paint that got spilled. It's actually the result of metabolic activity of biofilm bacteria growing on the rock face. They are taking metals from inside the rock and they're oxidizing those metals, iron and manganese, and which are very closely related chemically. And so the iron will produce the red color and the manganese will produce the black color. And they wind up sort of attaching to the rock and creating this varnish. Another thing that you see in the desert is something called cryptobiota. So this is the desert floor. This is in Utah, but you'll see it everywhere in the desert around the world. And if you look closely at what you're looking at here, they're actually communities of microorganisms. These are also biofilms. So they're a little bit like a lichen. So when you think about the desert floor, you think, oh, it's just sand and there's nothing there and it's all dead. Wrong. It's all alive. And they're all microorganisms. And they're interacting with each other. Here are a bunch of just pictures from Yellowstone National Park showing you different kinds of microorganisms, different colors. Remember pretty pictures? In hot springs. And then biofilms are also important with respect to industry. So one of the big problems with biofilms is that they grow where water is present and they wind up causing corrosion. So most of the rust that you see is actually caused by biofilm bacteria. They also cause problems if they're growing on things like a ship hull. So they impede the progress of a ship through the water. So, and Dr. Sauer will talk about corrosion, right? And biofouling. Okay, and then biofilms in health. So this is probably the first thing people think about when they think about biofilms, but much of what's going on in health we don't even realize is the result of a biofilm infection. So just as an example, one of the projects that I work on is hardening of the arteries, which we think of as being a lipid deposition disorder. Turns out that every patient who has hardening of the arteries has a bacterial infection in their arterial wall, and they're biofilm bacteria. So one of you guys will talk about that a little bit more.
I don't know if you're finished, but I have 2 questions.
Sure.
The first one, hold on a second, planning over there. The first over there, those that look like good and plenty, just one of them.
These?
Yeah, what's inside of it?
These are bacteria.
Yeah, but what's inside of it? Inside of one of those.
One of the bacteria?
Yeah.
So there's cytoplasm, and there's DNA in there. They don't have any membrane structures, so there are no what we call organelles. But all of the others, it's kind of like a bag of enzymes, but it's more complicated than that, right? So it's all of the enzymes and ribosomes and other structures floating around in this cytoplasm.
Do they give off waste?
Of course, yes. They need to take in nutrients, and they will eliminate waste by excretion.
Okay, but it's not like a contractile vacuole in a
No,
single-cell animal.
it
It's
is
just
not.
excretion. And the second question I have is, if you were a medical researcher and wanted to attack the bacteria, you gave those 3 examples, resistance and the other 2. Would you attack the biofilm? Would you attack the slime to try to isolate?
So that
the
is
bacteria?
one of that. Yes. So that is one of the, and I'm hoping that maybe Dr. Sauer or Dr. Marques will maybe address some of this. But that is one of the options that people have looked at. I've gone for the solution of you can't treat a biofilm. So what you want to do is convince the bacteria that they don't want to be in a biofilm anymore, and then you kill them. So, and I've been working on that, and although I don't have a lot of time to talk about that now, after the talk, if you want to stick around, we can have a discussion about that. Okay, and then this just gives you an idea of some of the different kinds of infections that are caused by biofilms. So acne, prostatitis, otitis, which is a middle ear infection. Some of you younger people may be familiar with otitis media. It's the #1 cause of hospital visits in children 10 years and younger. Burn wounds, cystic fibrosis. This is a metabolic disorder. It's a genetic disorder that leads to colonization of the lungs by bacteria that are in biofilms. Periodontitis, that's tooth and gum disease. Infected implants, like hip implants, for instance. Indwelling devices, like catheters. Non-healing wounds, particularly important with people who have diabetes and the elderly and chronic sinusitis. Many of us have sinus infections and we don't know how to get rid of them. Maybe all of us do. And those are biofilm infections too. Okay, so this is my transition slide to hand it over to Dr. Sauer.
Right, and he's a hard act to follow, but also left us a little time. But I hope that what you heard Dr. David say actually is that biofilms are everywhere. And people actually looked into certain environments and estimated that really more than 99% of all bacteria that you find in the water, in the soil, everywhere, are actually biofilms. So it's really the dominating way of how bacteria grow, not the way that we have been thinking about for 100 of years, meaning also that a lot of our treatment strategies are wrong. The antibiotics that we are thinking of will be treating an infection are based on cells growing planktonically, while really the dominant way of bacteria growing is as a biofilm. And as Dr. Davies mentioned to you, is they're actually extremely resistant to being killed by antibiotics. But what I would like to do is actually tell you a little bit of more specific examples of biofilms. Since bacteria are everywhere, so biofilms are everywhere, the question really that I want to sort of address is what are biofilms doing in the environment? What is their role there? What do they do? Where do you find them? So you saw a lot of rocks and water and soil, but I also want to point out that biofilms actually grow on every surface of plants. The leaves, the stems, the roots, inside the roots. And on the right side, you kind of see a few examples of where you see the root structure here, and the green is representing the biofilms that grows naturally on those surfaces. So, the question is, what do they do there? So this is my favorite example. Usually that would be me not watering my plants, but what it actually means is that biofilms, or the bacteria growing on those plants, will actually determine whether your plant is healthy or diseased. And there's an example, depending on, let's say, biofilms forming on the root, along the root structure, can determine whether your plants are growing very healthy and, grow up really fast and or look really like meager plants like mine usually on the right side, or even worse, wilt and die because they're actually being, there's fungi are getting into the root system and causing disease and killing up your plants. The way that biofilms usually protect and increase the health of your plants is primarily by a very simple mechanism. Is they're growing along the root surface and occupying every possible niche that is possible, and by doing so, preventing actually pathogens or disease-causing organisms to get close to your roots. By that, simply actually protecting the plants from attack, from disease, and other. In addition, while they're there, they actually do a few other things. For example, they're controlling pathogens and preventing them to come even closer by producing antibiotics on their own and releasing them. They can produce antifungals, for example. They can also prevent the plant from insect attacks, depending on where they are located. In addition to controlling pathogens from coming close, they also are known to stimulate plant health and plant growth, as well as crop yields, by producing, for example, nutrients or creating environments that actually is really beneficial for the plants. And thirdly, what they do is they do bioremediation, which is removing toxins from the environment or close to the root rhizosphere. Let's say pesticides can be removed by the biofilms that grow along the plant roots. It's kind of cool, right? But what I find even more amazing is that when you think of bioremediation being just happening around the root, they can actually do a lot more. You can think about pollutants in also a very different way of how biofilms can impact that. So bioremediation is a process that uses living organisms like bacteria to remove pollutants from the environment. It's really good for our plants. It's also really good for our planet. If you think about what mining and pollution can do to the environment, and biofilms in the environment can actually assist in cleaning up those sites. Another example for bioremediation is oil spills. You heard of Exxon Valdez, all these tankers, the Gulf being with oil spills. So bacteria, specific bacteria growing in a biofilm actually have been shown to clean up oil spills. And what you're seeing in this time course is a crude oil on the left side where bacteria started tearing on the outside of the oil droplets, start growing over time. And what happens, what you see here with the green indicating the bacterial cells, the hole in the middle is your oil, and it actually degrades the oil over time until nothing is left, and all you're seeing is the biofilm is being left that then will disperse and go look for a new oil spill. So biofilms in the environment can actually remediate or reclaim contaminated areas on a large scale. Unfortunately, it's very slowly happening. So it's not happening overnight. It takes a long time. The other fun part about biofilms is that they're not only helping cleaning up spills or help with bioremediation effort after mines are closing and contaminated the area. And I want to hint at, if you see what's sort of in the background, you see gold there. Biofilms also help creating or making precious metals. And in this particular case, gold. So biofilms are linked to gold nugget formation. And if you look really close at gold as it's found in the environment, you see this granular appearance of the gold, right? And if you zoom in a little bit, what you're seeing is the yellow one here is the gold nuggets, and the gray are the bacteria, the biofilms actually. If you zoom in further, it's actually the biofilm itself that helped create the gold nuggets by surrounding itself with gold. Kind of fun. Hurrying up. All right, contamination in biofilm. Dr. Davies mentioned biofilms help with or induce corrosion. What may be of interest to you is that corrosion not only happens on Earth, but it also happens in space. The first example of a biofilm being reported is actually from 1988 from the Russian space station Mir, where they realized that they had something growing or blanketing their windows that turned out to be a biofilm, and that biofilm continued to grow and started even destroying the quartz surface of that window. That's really not a good thing happening in space, I would presume. Right? And the biofilm started spreading actually to plastic panels, wiring, and corroded metals. And here are just a few examples of a door completely covered in mold, aluminum being damaged by biofilms pitting the aluminum. The top right is this fire suppression gauge that got actually corroded by biofilms. And what I find most amazing is that it's even went after the plastic wiring shown on the left there. causing electronic failures. And these two here on the lower right are examples of plumbing and solenoids for water valves that actually are completely covered by biofilms and contaminating the water on the space station Mir. So this is an overview from NASA pointing out all the different parts of a spaceship where now biofilms have been found, including water processors, showers, medical kits, now that's disturbing, air conduits, collection of humidity condensate, and actually humidity is a driving force of biofilm formation on dry surfaces like walls. Where urinals is a bit kind of predictable, but in other humid surfaces. So just to give you an idea of where they are, it's actually quite interesting. The other interesting is that now actually they even find biofilms on the outside of the space shuttle. And this was first actually discovered by the Russians where they found it. So how many bacteria are we talking about? How many organisms? So in 1990, they discovered 90 species of microorganisms. That includes bacteria and fungi inside Mir. 4 years, and this is only 4 years after launching, and they blamed it all on the Russians not cleaning up after themselves, and that number increased in 2001 when it was actually decommissioned to 140. So that's actually quite a lot, quite a number. They thought they could do better with the International Space Station. Everything that was actually launched into space was sterilized. So everything was cleaned up, everything was sterile, and guess what? So the latest numbers is that they now find there 94 different fungi as well as 96 bacterial species on that very nice and clean International Space Station. So today, not a single space station is clean. Previous and current space stations have all seen biofilms, all to the similar extent with the walls and the wiring being degraded. And Claudia, Dr. Marques, will tell you a little bit more about how these bacteria get up there. Are we still good with time? Probably not.
Yeah, no, that's fine. Yeah. Okay, my turn.
There we go.
I'll try to not to keep my 15 minutes. All right, so how did the biofilms get in the space station? How do you think? Any ideas? Yeah. Oh, well, right. Yes, you already know. He's my son, so he knows. Right, so the human microbiome, right? So we are really bringing the bacteria up to the space station. And we thought this was appropriate considering we're at Kopernik, right? And so microorganisms are in our body. So how much do you think we have?
Billions.
Billions. So how about comparing to human cells? All right, so imagine your whole body, right? Your whole biomass, and think of the percentage of bacteria that you might have versus human cells, okay? Your body is mostly bacterial cells. I think myself not as a human cell being kind of thing, more as a microorganism kind of person. Yeah, it's true. We have much more bacteria in our body than we have eukaryotic cells. And if you think about the genetic material we have in our body too, then the microbial genes make a large proportion of the genes that we also have and interact on every day. So the human genes are a very, very small amount of the genes that we have in our body. Okay, so think about us as a big microorganism with a big microbiome. So each of us has a different microbiome. What do you think can affect our microbiome? Yeah. Our diet, what else? Viruses, so disease, yeah, what else? Our moods. Well, actually, the microorganisms affect our mood, yes. Our environment, yeah. There's several things that some of them we already said. One is our age, our developmental we are developing, right? So when you were born, you have less microorganisms. As you get older, your microorganisms increase, and then they actually change. So, and they change over time with the age. Also, your genetic predisposition to have certain microorganisms in your body. Where you live geographically. So when you live here, for example, in Binghamton, you have sometimes dry weather and snow. Sometimes you have humidity in the summer. But if you lived, for example, in Brazil, in the Amazon, right, it's always humid and it's always raining. So the microorganisms that you actually have and that are part of your microbiome are different. Exercise can change your microbiome. Also, when you are born, if you are breastfed versus fed with bottle fed with milk in the bottle also changes your microbiome, because when you're a baby, you actually acquire your microbiome from your mother. So if you're breastfed, you have that microbiome. Hygiene, how many times you wash your hands, how many creams do you put, how much do you disinfect your household. Also, when you interact with humans and other animals, in this case, donkeys, for example. Thank you, David, for the picture. Yeah, so if you go to a pet zoo and you pet the animals, so you pick up their microorganisms, and eventually, if you're with them, particularly every day, right, so then some of the microorganisms the animals have are part of our microbiome too. What we eat, it's very important for our microbiome. We really are truly what we eat too. And also medications that you take, including not medications, but also vitamins and herbal supplements that you might take. Those affect your microbiome. Okay, I'll try to be quicker. All right, so is it important? Yes, it is. One of the things that is really important, and I don't know if you heard about it, but in the last 2 decades, a lot of people are talking about gut microbiome a lot. And our gut microbiome is really important, but also because it actually talks to other organs in our body. So it can talk to our skin, it can talk to our, for example, mouth, but it also talks to our brain. So that's what they call now the gut-brain axis. So the bacteria in our gut actually produce chemicals, as Dr. Davies talked about before, and those chemicals are actually transported through our bloodstream, and eventually some of them will reach our brain. So they actually talk to our brain, and they affect how we behave, what food we want. So have you ever, for example, craved some kind of food? Oh, today I want chocolate. And then the next day, oh, I want to eat a pickle. Or, I want to eat cheese now. Or I need something really, really dry. Or I'm really thirsty, right? All that stuff that we have, that we're craving, a lot of times is our gut saying to our brain, I need this. Or our body, yeah.
So there is no free will?
That's a good point. I don't know. Actually, my original title was, Are We Really in Control? But I changed it. It's just you still have the power of will, right? But a lot of times your bacteria actually talk to your brain, and your brain is telling you, I need this, which is kind of cool. Anyway, but your gut microorganisms affect the food you're intaking. Also, they've been related to some of the chemicals to the presence of Parkinson's disease, and even relating to Alzheimer's now. Also to cardiac disease, whether you're happy or not. Okay, so if you keep your gut happy, you normally stay happy. And relating to your stress affects your gut, which eventually will affect your brain. And social and cognitive behavior too. So, but we are in the Kopernik. So what happens to our biofilms during space travel? You like that, Drew? See? All right. So when we want to be astronauts, that was him when he was 4, right? And he wanted to be an astronaut at the time, as you see there. So what happens? So I'm going to give you one example of a simulation that was done, which was Mars 500. And Mars 500 was done in cooperation between Russia, Europe, and China. And it was a controlled simulation of an expedition to the Red Planet. So they really simulated the Mars environment. So there's a Mars lander module, habitation module, some medical module where they actually took like blood pressure, you know, and oxygen availability in our body, body mass, body weight, right? And they took blood samples to see what we had in our blood. And then there was the utility module. So they had a gym, a greenhouse, storage lavatory, lavatory. And they also had an area that was similar to the Mars surface. So for them to move from this area to the Mars surface, they actually had to put their spacesuits like those on that you can see there. Okay, so this was with a crew of 6 people that spent in this environment alone, just the 6 people, no contact with the outside, for 520 days in isolation. This is the living room. Okay, so, right? That's pretty cool. Okay, so what happened? So to this 6 crew, they took samples on the beginning, once they first entered this area, and then at the end, just before they were leaving. So they took samples of at day 7, 14, 21, 30, and 45, the first 45 days, and they considered that the healthy, so when they started, and then they took it at the last 100 days, 30 samples from the six astronauts. What did they find from beginning comparing to end? The astronauts lost total body mass, lean mass, BMI, those are all decreased. They also had a disruption on their glucose metabolism, okay, indicating possibly diabetes, and also increased of immune response. So their immune system was kind of dysregulated. But then they also looked at the fecal matter, so what they were excreting with their feces, to check on their microbiome. And what did they find? So if you look here on this side is just how much the bacteria changed, the fold change. And on the bottom, they're either known bacterial species, firmicutes that usually live in our gut, versus unknown species. Our gut has a lot of microorganisms. There are billions and billions that a lot of them we don't know yet what they are. So if you look at the blue is the early, so the first 45 days. And you can see that there's a lot of known organisms and some unknown. Now look at the late period, the last 100 days. None of the ones in the early days are present. They all changed, right? So these were the most prevalent ones, and now those are not the most prevalent ones. The ones that are most prevalent in the last 100 days are, a lot of them are unknown, and the ones that are known are different from the previous ones. So their microbiota in their gut actually changed. And a lot of times that causes disruption. Good point. They change because the oxygen conditions are different. They change because the food is different too. So a lot of food that they eat will be lyophilized food. They change because of radiation also, and also the gravitational environment that is different.
And this was 6 Russians?
No, there were 6 people. I'm not sure if they were all Russians. But it was a combination between Russian. Yeah, Russian was not the major, because it was Russia, Europe, and China. The US actually has a station that is simulating Mars in the desert in Arizona. Right, and they did this not, so another example of that is that this was also correlated to previous studies that they had done to astronauts that traveled into space, that went to the space station. And if you consider the health gut microbiota from the beginning, which is, you know, your system is all great and you're happy, they found that astronauts, when they come back, they have disrupted microbiota, particularly in their gut, and due to the microgravity and the radiation of space travel, and also their food and their conditions. So what happens is that they have diabetes, altered gut bacteria that causes diarrhea, suddenly they have inflammatory bowel disease, and then they have bone loss, right? And some of them come back with cataracts starting, and also some heart diseases. And they're not entirely sure, but they think that this dysregulation of the microbiome may also cause cancer in some cases. So we're trying nowadays to actually restore this disruption. One way of doing that is to use probiotics. And now it's pretty in fashion for people to say, oh, we should eat more probiotics, right? Different food that already has bacteria. And we know that probiotics can help you maintain the good bacteria in your gut because it displaces the bad bacteria, and also improve your heart condition, definitely because the bacteria goes back to normal, then your diarrhea goes away, and also help with allergies and your own mental health too, because again, the gut is talking to your brain. And on that note, I'm going to go to the next one, Karin. Right, so I should say that there's also other methods for you to protect from the disruption, and we can talk about that if you want to. But we just wanted to say that overall, I hope that you take that biofilms are all around us. They're a part of us, a big part of us, and of our environment. We live with them, and sometimes we do not like them at all, but we cannot live without them, okay? We believe that, well, definitely I do believe that without microorganisms, we could not be here today. Okay. I need to come closer. There you go.
For some reason, I'm left with finishing up to say, like, the question is, you know, we are working at Binghamton, so what do? We are actually part of the Biofilm Research Center. We are now 8 faculty members that work there, and these are our students. And us. So the research that we cover ranges from basic mechanistic studies, understanding how biofilms form, all the way to how to control biofilms. We are looking at just basic understanding of how one species makes a biofilm, to how biofilms interact with the host, or how the host responds to one or more biofilm type. So it's quite the range of different topics that are being covered by us there, and we would thank you by now. Actually, now we want to thank you for your attention because we've taken way too much of your time, but thank you.
Only 12 minutes, so
Only 12 minutes.
All right, well, we do have some questions, and so I don't know who wants to answer them, but actually, do we have any questions here in the house? Okay, we got another one here, so.
So on the Exxon Valdez, when they had that oil spill, who determines what kind of bacterium that you're going to introduce to try to dispose of, and how long would that take?
That's an excellent question. So Exxon Valdez, I believe they started out with whatever was there naturally and enriched it, and it resulted in the first one and only native bacterium ever being patented. Pseudomonas putida, and it grows very slowly on oil. Oil is very complex. It takes a long time. I think bacteria may divide once a month, so it does take a while, but it happens naturally. You just need enough of the bacteria. So now they're actually calling something like oil zappers. It almost like you can buy it in bags and apply it to different areas, but it's always the same bacterium basically. Exxon Valdez, I think it's cleaned up by now, unless it's settled to the ground. It's but it took a long time. Mostly cleaned up.
Within about 15 years, most of it had already
But
been removed.
so they now know you can go in and apply bacteria to and encourage biofilm formation to clean it up, and they actually have shown those where they applied it versus where they haven't, cleaned up a lot quicker. So.
Another question back here.
What were they applying in the Gulf?
Nothing.
What would they apply in the Gulf?
What did they apply?
nothing.
I think they put just detergent in it to
So-
have it settle.
Yeah, actually, I can answer that question more specifically. Yeah. So it turns out that one of my PhD supervisors was the inventor of the only patented organism used in bioremediation. And that organism was released. His name is Ananda Chakrabarty from University of Illinois at Chicago. And the organism was actually allowed for release in the Gulf initially. So it was used primarily in the Gulf. And it was genetically engineered. No. The problem was that native microorganisms outcompeted the introduced species, so they were involved in remediation for a short period of time until they were outcompeted and then lost. And just to sort of add on to what Dr. Sauer was saying with Valdez, in Alaska, what they wound up doing that did work wasn't so much bringing in new microorganisms, it was actually feeding the existing organisms with, I always like to call it just steak and milkshakes. So things that bacteria like to eat, and that help them to then also digest the oil.
We have a couple, one or two comments from and a question from the chat. Again, we talked about that. Tubik is in fact going to get a new sponge as soon as this talk is over, and but they also ask question: how do I not become a germaphobe after this? Bacteria are everywhere. Any reason not to be scared of this fact?
Sure, bacteria are everywhere, but they're not always harmful. It's all about a balance, right? They balance themselves out. We have bacteria. We're with them every day. I'm not a germaphobe.
I think it's important.
It's important.
To mention that the three of us all obey the 5-second rule.
Yes, definitely. But, it's fine.
So you're covered from the outside to the inside with bacteria, right? And so even if you lick your skin and you have a tequila shot, you're actually, ingesting a lot of bacteria. But here's what is really more important is if you were to think about being completely sterile, no bacteria on you and in you, you wouldn't be able to function. You would get sick really quickly. You wouldn't be able to digest your food. Your immune system would crash. You would start developing allergies or not develop in general well, like you would today.
Also, the bacteria that you have in you or on you are usually good bacteria, and they fight the germs that we encounter on a daily basis. And that's why you want a healthy microbiome, is because that microbiome can fight the bacteria that cause disease. So no need to be a germaphobe.
All right. So Lindsay, our friend in Adelaide, Australia, asks, so microbiome and jet lag, it's a real thing.
Oh, probably, yeah, definitely. I have that all the time when I go to Portugal to visit my family. Yes, jet lag is definitely a thing with bacteria, yeah.
And then Mike asks, are you involved with NASA for assisting for astronaut long-term health in space?
We're not our group particularly, but there are groups that are doing that, and I think Dr. Davies knows some of them more than I do.
Should I tell my story?
Sure.
So,
Tell me your story about NASA.
I have a very short story, unfortunately, about my interaction with NASA. So I did one biofilm experiment in space on the space shuttle, and so we developed a device where we could grow biofilm bacteria and look at what on the space shuttle and then potentially in other locations like the space station might actually be responsible for culturing these bacteria that are growing on things like the outside window of Mir. Unfortunately, the experiment was done on the Space Shuttle Columbia. And it blew up on the way on reentry, and my experiment package wound up somewhere 6 feet underground in Texas. And we were never able to get results from that, so, yeah.
Actually, one comment I'll make is that, again, we do a fair amount of engagement with NASA, and I know that a lot of the work that the astronauts do on the space station is literally cleaning the space station constantly. It is so important, and it clearly, the early work or discovering those microbiomes, it's absolutely clear that if they're going to try to stay even close to being healthy, they need a really clean environment. So, any other questions here? Oh, another one here.
Speaking of space-bound biofilms, have we found any that have survived re-entry yet on the outside of a shuttle? No? All right.
So, they only have seen them on the outside, those that stayed up there for extended period of time. So I think upon re-entry, I'm pretty sure they get
They get
burned.
just on the outside.
Yeah.
The ones on the outside leaked out
Yeah.
along the gap where the windows were. And so they were just right outside by the border of the window.
However, you do find bacteria and fungal spores at 10,000 feet or higher. So it's possible they may be traveling up into space, but all the biofilms they've seen that come through the windows, and that's where they have been sampling them from the outside. It's kind of, but they're live, which is even more astonishing.
All right, unless there are any other questions, I thank you, all three of you, very much for just a number of people commented how fascinating the program was. So thank you for bringing biofilms to our attention. And so when they say employees must wash hands, that means more than just the employees. Everybody must wash their hands. And go buy a 10-pack of sponges, I guess, at this point. So especially for those people who are maybe haven't been here in a while, or this may be the first time, we always have a rule here that you can't leave Kopernik until you look through a telescope. Unfortunately, tonight, but we'd be happy to give you a tour of the scopes, and we can show you some of the images that we've taken with our scopes. So it's 8:30. Well, in 7 minutes, we'll start out in the lobby. Anybody who wants a tour, we'll do that. And thanks again.